All right, so happy Monday, everybody. Welcome back to Paleo class. So we're starting a new unit here. We're going to be talking for the rest of the class on origins and others. So we're going to talk about origins of life, how life first came to be. Probably we're going to discuss it. Obviously, there's a lot of uncertainty there. Going to get into early evolution, which we talked about a bit. Going to talk about some quote-unquote vertebrates, so things that are almost vertebrates. Basically, it's just kind of a repository for some of the organisms that we didn't get a chance to talk about in lab, so like Graphite and condensed and things like that. And then we're going to talk about actual vertebrates, so we're going to talk about fish and mammals and us, and going to get into trace fossils. And then the last week of the class is going to be plants, Palynology, study of pollen, and Paleoclimatology. So we'll talk about how we can read past climates. We talked about how important those were to the development of life, and we're going to talk about how we studied that. So this is where we're going. Roughly covers chapters 11, and then jumping to chapters 18 through 20. Chapters 12 through 17 we've already kind of covered in the lab, or will cover in the lab. So we'll basically have gone through the whole textbook by the end of this, focusing more on certain parts than others. Before we jump into it, let's take a look at some announcements. Oops, let me pull up my highlighter here. So exam 3 is over. There's 2 weeks left after this, so we're coming into the home stretch here. And just remember, if you haven't been participating in the discussion boards or you've been missing some labs, please come talk to me. We can probably work something out, okay? So let's get into it. So if we're going to talk about the origins of life, let's start with the origins of our planet. So the first geologic eon is the Hadean. So Earth formed about 4.5 billion, so like 4.54, 4.56, somewhere in that neighborhood, years ago from, and again, it's accretion from smaller fragments. So gravity kind of brings small pieces together, they smash together, they make slightly larger pieces. Those larger pieces smash another larger pieces, and it just gets bigger and bigger and bigger over time. So that's called accretion. So like think about if you're going to go build a snowman, you start with a snowball, you pack another snowball, and your snowball gets bigger, and then you got a nice base for your snowman, and you do it all over again. That's how the planet was formed. Gravity kind of pulls these things together, and it just gets larger over time. So this is kind of an artist's rendition of what that might have looked like. And obviously no one was there to see it, so it's just a guess. But early Earth was just scorching hot, and you know, fire, brimstone. It strongly resembled the conception of Hades, hell. The atmosphere was toxic. We wouldn't be able to breathe it. If you were transported back in time to the Hadean, you would suffocate and die. It's rich in ammonia, rich in methane. And the oxygen that we need to breathe, there was less than 1% of free oxygen. And at least initially, there was no surface water at all. So if we were transported back to this environment, we really wouldn't recognize it as Earth. It's this hellish landscape that's completely inhospitable to us and most other forms of life. Originally, this was kind of thought to linger for, you know, hundreds of thousands of years. Hundreds of millions of years to maybe even a billion years. That's kind of getting pushed back and back and back, that the Hadean, this actual hellish landscape probably wasn't quite as long as we originally thought. And one of the first things that kind of ends this is the oceans. So once Earth cools enough for liquid water, so initially when you're in that hell-like landscape with molten rock covering the surface of everything, and that methane ammonia atmosphere causing a strong greenhouse effect, water can't exist in liquid form. So one of the big diagnostic features of our planet from space is we're called the blue marble, because over 70% of our surface is covered in liquid water. And then you can even see the vapor water in the clouds. And then if you look on the ice caps, you have solid water. So one of the cool things about our planet, one of the things that probably is necessary for life, is that while we exist just the right distance from the sun, that water exists in all 3 phases at surface conditions. And that's not true everywhere in the universe. It's not true everywhere in the solar system even. It's that particular condition for life is probably not unique to us, but we've never really observed liquid water elsewhere. We probably have remotely observed it, but we've never directly observed it. So where did it come from? Well, water came from volcanic outgassing. So just H2O water that was in the mantle was kind of belched out from volcanic eruptions. And that was some of the water. Some more water arrived from comets. So remember, comets are basically just these ice chunks, these dirty snowballs. So like rock and dirt and particles kind of glued together with ice. They collided in Earth, the ice melts, and we get a little bit of water. There's a lot of debate about how much water exactly came from outgassing versus how much came from comets, but it's some combination of the two. And basically, so water was in the air as steam, and as the Earth cooled, that steam condensed into clouds, and then it rained out as liquid water. It filled in all the little low spots, and we started getting what eventually became the oceans. And it probably took millions of years, tens of millions of years, maybe hundreds of millions of years of raining for this to happen. And then our first oceans were formed. And one thing that's becoming increasingly clear, especially through looking at zircons, is that the oceans probably formed relatively early in Earth's history. So how do we know that? Well, zircons are these really rugged, really resistant minerals that they long outlive the rocks that they were initially formed in. So a zircon is formed in a particular rock. That rock is exposed at the surface. That rock is eroded, destroyed, and recycled in the rock cycle. But the zircons are preserved because they're like little time capsules. They're very rugged. They are not broken down. They're very hard to kill. And so they keep kind of getting recycled over and over and over again. So there are actually no rocks from the Hadean period. Nowhere on Earth's surface that we're aware of are there rocks from this initial Hadean period, but there are zircons that date. So these are like time capsules of this earliest chunks of Earth. The oldest one is, I think, from Australia, about 4.4 billion years. So only 100 million years after the formation of Earth. So already at this point, we've got solid rock, or the zircon wouldn't be present. And there's evidence pretty quickly from the chemistry that water is present fairly soon too. So we didn't have to wait until like 4 billion years for the oceans to form. So, and those dates keep kind of getting pushed back and back and back, where the Hadean period, where Earth was very hell-like, is getting kind of shorter and shorter and shorter. But another thing that's interesting in these zircons is that some of them preserve graphite, and that graphite, remember graphite is a carbon mineral, that graphite has a low carbon-13 ratio, and we talked a little bit about carbon fractionation. So when organics take in carbon, they preferentially take in the light carbon-12, and so having graphite carbon that's low in C13 is often indicative of biological activity. So all the way back to 4.1 billion years, there's some evidence that there's some sort of life on Earth. We'll talk about when the evidence gets strong for life on Earth next time, but it looks like basically as soon as water is present, life is present shortly thereafter. And these things keep getting pushed back. So it's pretty interesting. So how did life come to be? Well, that's always kind of puzzled us, and it's a very interesting question. Obviously, one of the most important questions is kind of like, why are we here? What are we doing here? How did we get here? The initial guesses was like spontaneous generation. So it was initially thought that life was just able to kind of spontaneously generate out of thin air. So like, for example, with rotted meat, so you have meat, and meat just kind of sits there, nothing apparently interacts with it, and then all of a sudden, like, maggots just start magically appearing out of the meat and develop into flies. It was kind of like a magic trick. These flies just came out of nowhere. They spontaneously generated. Or like there was an idea that fish are maybe spontaneously generated from mud. or mice are spontaneously generated from grain. So it's not that they were like, initially, they like magically sprung from the mud or magically sprung from the grain. And then Louis Pasteur kind of did an experiment, one of these experiments here, but he got some like broth, so, soup basically. And if you leave soup out on the counter for weeks and weeks, I don't want to think what's going to happen to it, but it's not going to be pretty. But he boiled the broth to sterilize it, and then he put it in a container where it was locked away from any influences, and it didn't spoil, it kept. So he sort of invented this idea that, life doesn't just spontaneously generate from something, there's some source to it, and if we can sterilize something and keep it segregated, then it's not going to become polluted. So that's where kind of pasteurization came from. So if it wasn't spontaneous growth, then what is it? Well, it's very difficult to solve, because on Earth today, under current conditions, life comes from life. So it's kind of like a chicken or egg thing, which came first, the chicken or the egg. Life comes from other life in all cases here. So one thing that's a little bit different is that today on the modern Earth, life is ubiquitous, it's everywhere. It's in a lot of places that we didn't even expect. A lot of deep drilling now is starting to find these microbes deep down in the earth, away from any kind of logical nutrient source, feeding on rocks and chemicals. We didn't think that would happen, but we're starting to see that pretty much everywhere we look, life is kind of finding a way. So pretty interesting. But now, in the present conditions, because life is everywhere, and life is greedy and it's wanting to use up the nutrients, as soon as any kind of organic building block becomes available, or any kind of nutrient becomes available, the existing life quickly gobbles it up. So there's no way for that stuff to kind of accumulate. And so this is kind of an exception to like the present is the key to the past. If we want to look at the past conditions for how life formed, those conditions probably don't exist on Earth anymore, because everywhere on Earth life is already there. So life is already consuming all those basic building blocks, or at least that's kind of the idea. But again, very early on Earth, there was no free oxygen, so there was no way to oxidize these particles that are just kind of hanging out. There's no hungry life forms kind of gathering them up. And so there was this idea that Darwin kind of coined, this primordial soup. So this nutrient-rich soup, this warm pond of just organic material that eventually life just kind of sprung from. And so we know that from organic chemistry, if you've taken an organic chemistry class, these are the kind of basic building blocks of organic chemicals. So you have hydrogen and carbon, you put those together, you get hydrocarbons. So like methane is a hydrocarbon. Carbohydrates and lipids, if you add oxygen to the carbon and hydrogen, so like C6H12O6 glucose, that's a carbohydrate. That's one of these things. If you add nitrogen, you can start forming amino acids and proteins. If you add in sulfur, you get even more different amino acids and proteins. And then when you add in phosphorus, you're able to get nucleic acids like RNA and DNA. And so if you've taken organic chemistry, there's kind of this acronym SCHNOPS. SCHNOPS. It is basically with just these 6 elements, you can form something like 98% of organic compounds. So organic chemistry is just kind of like rearranging these 6 things in various different ways to get all organic compounds. So again, thinking back to that early atmosphere, methane's a hydrocarbon, that's present, ammonia is present, sulfur is being belched out of the earth. We have these things present. Nitrogen's free in the atmosphere. These organic building blocks are here. The elemental ingredients are there. And they're kind of, again, the theory is that they're kind of floating around in this primordial soup. So how do they start like combining into more complex things? Obviously, like hydrogen and carbon alone are not life. Even methane alone, a hydrocarbon, is not life. Glucose, sugar by itself, is not life. How do we start making actual life from these building blocks? So one of the key breakthroughs here was, so Stanley Miller in 1953, he made this kind of neat little apparatus here where he basically had simulated ocean water, so just sterile ocean water, salt water. He had a valve in here where he could pump in something that resembled the ancient atmosphere, so carbon dioxide, methane, ammonia, hydrogen, and then that kind of moved through here. So it would be like steam from the heated water in here. It's going to condense as it expands. You've got all these other or compounds present from the atmosphere. And he starts zapping it with lightning, or zapping it with electricity to kind of simulate lightning. And then the water vapor condenses and it gathers here in the trap and sort of goes through this loop. And so you're basically exposing seawater to the ancient atmosphere, you're zapping it with lightning, and then it's cooling and it's condensing, and you're seeing what happens. So what was kind of cool is that in a few days, the clear water that was sterile salt water, it started turning brown. And then after about a week, it was like this thick organic like goop. And when he analyzed that goop, he was able to produce like 20 different amino acids and a couple other organic molecules. And so this is basically, he recreated the primordial soup. From sterile seawater and inorganic compounds in the atmosphere, combining those together, you're able to get organic material. So this is kind of the first steps towards like abiogenesis, the genesis of life from inorganic material, which is in theory how life originated. And so this experiment's kind of getting there. But again, amino acids are more complicated than proteins, but they're not, or sorry, amino acids build proteins, but they're not life. Organic molecules are not life, they're not living. So we're still not quite there yet. Another idea that's not abiogenesis, although ultimately it probably has to still be abiogenesis somewhere else, it's this idea called panspermia. So by analyzing different meteorites, we've been able to find 74 different amino acids trapped in them. So looking at rocks kind of flying through space, a lot of rocks have organic components to them, organic compounds. So we see that organic compounds are being produced elsewhere in the universe. So did the organic material that we find on Earth kind of hitch a ride here on a meteor from somewhere else? One thing that Stanley Miller's experiment kind of proves is that making the basic building blocks isn't really the problem. So whether the building blocks came from primordial soup or whether they came from asteroids, that's not really the limiting factor here. And so it's very easy to kind of build these building blocks. So that kind of makes this theory a little bit unnecessary. The building blocks were already here, and it's relatively easy to build them. So it's kind of a cool idea, but it's probably really not completely necessary. So let's talk about life. So I said amino acids aren't alive, but what does it mean to be alive? So we talked about this a little bit when we first started up with the lab. What is life is a very difficult question, but there are some kind of broadly agreed upon criteria here. And one is that something that's alive is able to maintain homeostasis. So it's able to maintain an internal environment that's different from the external environment. It's able to kind of maintain, say, a constant temperature or a constant salinity or a constant pH. It's able to modify its internal environment to its desired characteristics, even if the outside environment is changing or different. There has to be some sort of organization, so there has to be some kind of structure to the cell. Specifically, probably there has to be a cell membrane to sort of isolate the cell from everything else. There needs to be some sort of growth, so the organism needs to kind of increase in size. It needs to have some way of either capturing energy, so like eating, or making energy, like photosynthesis or chemosynthesis. It needs to be able to react to external stimuli, and that doesn't need to be as complex as like animals responding to external stimuli. But if conditions change, the organism needs to be able to respond in some way. Maybe not good enough, but they need to try to respond at least. They need to be able to reproduce, so to make new individuals. and they should kind of change over time. So amino acids and organic molecules, those are building blocks of life, but they themselves do not meet most of these criteria. And so most people would agree that they are not actually life, they're not alive, but they are the Legos that we're going to put together to build the sculpture of an actual living thing. So how do we get there? How do we build more complexity? So again, this idea, abiogenesis, generating life from these inorganic compounds, building into these organic compounds, and then kind of combining them together even more complex. How do we do that? So basically, if you look at proteins, proteins are amino acid plus another amino acid combines to make a dipeptide. And if you start smashing a whole bunch of those together, you get a polypeptide. So poly means many, so many peptides. Then you get a protein. So if we're able to make amino acids relatively easily in the primordial soup, to make proteins, we have to combine those together somehow. And they have like very specific structures. For carbohydrates, you take a simple sugar, combine them together, you get a disaccharide, 2 sugars. Combine those together and you get a carbohydrate, polysaccharide. Again, poly is many. So again, you start with these simple building blocks. You have to arrange them together in a particular way, and then you get these kind of like more complex molecules. Same thing with nucleic acids, like DNA and RNA. You got to sugar some of those bases, ACGT, yeah, and then you combine them together. And then for lipids, lipids are kind of relatively simple. You have like a hydrophilic, usually like an alcohol or something, and then a hydrophobic kind of fatty acid tail. And they kind of orient themselves with the lipids, this hydrophilic on the outside and the hydrophobic on the inside. And they kind of like can wrap themselves up in like little globules. So again, to make these complex molecules, you're just basically assembling simpler stuff. It's easy for us to get the simpler stuff. It's a little harder to bring all these things together in the right order and in the right quantities to build more complexity. So how do we do that? Well, one of the ideas is that you need something that's like called like a proto-cell. So Oparin, a researcher, had this idea of called coacervates, I guess how you say it. And Fox had a very similar idea of proteinoids. And basically what they were is they're droplets of lipids, and they have nucleic acids kind of trapped inside. If you've ever dropped oil into water, you'll see that just the fact that the lipids in the oil is hydrophobic, it sort of forces these little spheres, these little globules. And so inside those, you can actually kind of capture proteins or polysaccharides, and then you get kind of these like self-enclosed globules of organic material. Still not truly a cell, but we've got like an outer membrane, a very simple membrane, and they can actually selectively absorb certain materials. So they can selectively absorb organic compounds, and they can selectively remove toxins, waste products. And so it behaves similarly to a cell, but it's not quite there yet. They can even grow as they accumulate more material, and they can even actually bud off to kind of reproduce, not really copies of themselves, but another proto-cell. So it feels like we're getting closer. We've got simple organic molecules present. If we're able to assemble them in some way, we'll start getting there. This is one way to kind of concentrate them and sort of protect them from the outer environment. Now we need to start kind of assembling them into bigger ordered pieces. So how do you do that? Well, one way that we build complex things is we use like templates or scaffolds. So like, for example, these are a couple common examples that we interact with. So like, if you're gonna play Jenga, you don't just stack all the blocks up by freehand. I mean, you can, but it's difficult. So we have this template that comes in the box that you kind of shove the pieces into, you press it in there, and it kind of it builds this more complex tower from these simpler blocks, and they're ordered in a certain way. And now instead of individual blocks, you have the Jenga tower assembled. Another option is like pool balls. So you use the triangle to kind of rack up the pool balls in a certain shape. That's a template. It'd be very hard to just rearrange all the balls by hand to get that triangle shape, but you have this template. Or like in dressmaking or woodworking, if you want to build something sort of complex, you have a template first, and you just kind of mass produce the pieces, and you put the pieces together, and you already know they fit because you've made a template. Another good example is the square peg and a round hole. There's sort of this like filter here that doesn't let you put the pieces together any other way than the right way. And so when organic chemists are working with these building blocks, they'll often use catalysts to kind of speed up reactions, and they kind of like force like intermediate steps or sort of force things to kind of line up, and it speeds things up. One thing that might have helped with this transition from these simple compounds to these more complex compounds is using inorganic scaffolds, like say minerals. So remember, minerals are inorganic compounds that have like a set crystal structure. That set crystal structure might have served as early templates for forming organic structures. So pretty interesting. So one way that this could happen is with clay minerals. So clay minerals, think about your like biotite and muscovite in intro, where you tear apart the sheets. Very satisfying, very fun. They're inorganic sheet silicates with the one cleavage plane. And they're formed from these complex repeating structures. And so there's like a crystal structure here on one sheet, one layer. That same crystal structure is repeated on the other layer. And then in between, there's a gap. That's the cleavage plane. That's where the sheets would come apart. In that gap, organic molecules in water can be attracted to those inorganic sheets above and below, and they can kind of be lined up by the charged particles on the inorganic sheets. And so this process is like so efficient that when new clay minerals are growing from old clay minerals, they can even inherit like mutations from the original parent lattice. If there's like an impurity in one of the layers of the clay mineral, when the next layer grows on it, it'll actually repeat that mutation. So that should sound kind of familiar, because it's kind of almost the way that like DNA works. One of the base pairs gets like corrupted or like added or subtracted or combined or misaligned. When you split that and you reproduce it, that mutation might end up in the next generation. So sounds pretty familiar. And this is inorganic molecules doing this. And the idea here from this book, Clay Minerals and the Origin of Life, is that on these microscopic layers of inorganic clay, there's already this like rigid structure, this rigid repeating structure, and the organic molecules kind of just followed along using that inorganic as like a scaffold or like a template to put themselves together in a repeating pattern that sort of mimicked it. So this is one of the ideas of how you get from something that's relatively simple to something that's seemingly impossibly complex. There were already these very complex structures existing in nature with inorganic chemistry. It's possible that organic chemistry used them as kind of like a substrate to kind of use as a platform for forming the way to kind of like build themselves into more complex chains. So clay minerals is one of the proposed mechanisms of this. To my knowledge, it was the first. Another one that could possibly work is zeolites. So zeolites are microporous aluminosilicate minerals. Because they're microporous, they're able to absorb things pretty well. So they're used commonly as adsorbents or catalysts or filter media. So if you think about like clumping cat litter has zeolites in it. That's what makes a clump. It's found in water purifiers, so like fish tank water filters, and also in detergents because it absorbs organic stuff. That's the whole point of a detergent. As organic molecules are kind of pulled from water in this filtering process, as they flow through the microporosity of the zeolite grains, they align along the surfaces of the zeolite. So again, the zeolite is a mineral. It has a inorganic repeating structure to it. And if these organic molecules adhere onto that repeating structure, they're going to form their own repeating structure. And again, this is a way that you can assemble something more complex from something simpler. You've already got an existing template, an existing way of sort of organizing all these minor components into something a little bit more major. And then the last one that's been sort of proposed is pyrite. So pyrite, we know it as fool's gold. So fool's gold, pyrite is iron sulfide, so FeS2. It forms in reducing environments, so where oxygen is not present. So instead of rust. So again, when we think of like geology, a lot of times we'll go an outcrop and we'll see like these alternating like red and green bands. So the red bands are quote unquote red beds, usually indicative of terrestrial deposition, where we have oxygen present, you have rusting conditions, and then you go to the green band, and that's something that's present in a reducing environment where the oxygen has been used up. So like some sort of anoxic bottom waters or a swamp or something where there's a lot of decay using up the oxygen, you'll get like a green layer, and you might see that kind of switch back and forth through the outcrop, making these red and green bands. Pyrite is present in the reducing environments. One place on Earth now that we find a lot of pyrite is at these black smoker hydrothermal vents. So these are a relatively recent discovery from deep sea submersibles. I think they were discovered in the late 70s. We didn't even know they were there before. But basically, you're at like a mid-ocean ridge or a hot spot or somewhere where there's a lot of magma deep down underneath the crust. Seawater kind of enters through cracks in the rock or pore space in the rock, and it comes in contact with the magma or nearly in contact with the magma. It heats up, it rises, and you get this expulsion of this hot water with all these nutrients and all these minerals dissolved in it. So that sounds pretty good for life, right? There's lots of heat, there's lots of minerals, and what we see around here is that these black smokers support these like really unique ecosystems, these like isolated pockets. In general, there's not a lot of life down on the deep sea floor. These are like these oases of these very diverse ecosystems locally around these vents. And one of the more intriguing things about these is that there's a lot of these Archean chemosynthetic bacteria that live down here. So we talked very briefly about archaea when we were doing the protist lab. So archaea are more primitive than protists. They're the most primitive life form on Earth, which suggests that they're probably the first life form on Earth that developed. And we find them in great numbers around these hydrothermal vents. Obviously, they exist elsewhere as well, but one of the best places to find them is along these vents. And so did life originate at these vents? Is this where these Archean bacteria originated? Kind of an intriguing idea. But again, the idea is basically the same as these others, is that pyrite crystals, inorganic, repeating mineral crystal lattice structure, that repeating lattice acts as the polymerization scaffold. It's the template that the organic molecules could use to build from the simple building blocks up into the larger stuff that builds actual true quote unquote life. So we have progressed from the building blocks. We're starting to build more complex stuff, but we're still not alive. How do we take that last step? So recently there was a proposal of something called RNA World. So it's very easy to polymerize the basic molecules into complex ones. Like it's pretty simple now to kind of explain that you build amino acids with the Stanley Miller experiment. So those are just going to happen through the combination of the ancient seawater, the ancient atmosphere, and some heat. You're just going to naturally, spontaneously get amino acids. You start combining amino acids on some sort of template or scaffold, and you have proteins. But how do you get from proteins to more complex stuff like the messengers of genetics, RNA and DNA? So in order to have life, you need to have reproduction. You need to have some mechanism of genetic information, some way of carrying information from one generation to the next. And one thing that was interesting is that so certain types of RNA, they're called ribozymes. Ribozymes because they actually occur in ribosomes, the cell organelle. They serve as both genetic code, but they also serve as catalysts. So they're kind of like these multitaskers. And so this idea is that like DNA is fairly complex. It's kind of unlikely that you would make a leap from amino acids to proteins to DNA. This RNA world is the idea that DNA wasn't really present yet. Organisms used RNA first for their genetic material, and only later was it replaced with DNA, and possibly a couple like intermediate steps in between there. But basically, so summarizing back, we start with the abiotic stew. We build kind of simple organic molecules, assemble those into RNA nucleotides, and then we assemble that into RNA itself. And then RNA helps in protein synthesis. It starts building these things. And from there, we can kind of understand how complex life would be able to form. You notice all the question marks here. This is kind of the stage that has the most uncertainty to it. How do you make that jump? How do you go from proteins to RNA and DNA, where you can actually get a complex living organism? There's still a lot of questions about this, but you can kind of see how this might happen. And obviously still a lot of research that needs to be done, a lot of probably fairly difficult conversations that need to be had, but pretty interesting nonetheless. So this is kind of how life might have formed. Obviously, we don't know. We may never know. We might never be able to fully reproduce this process in the laboratory because, again, the idea that the present is the key to the past, current conditions are very different. So how could we make an experiment to generate something that the conditions don't exist? The Stanley Miller experiment is kind of like pushing us that way. Maybe we could do some more experiments like that, but we're kind of limited by our current environment and the fact that life already exists. So if the present is the key to the past and life already exists, it's very hard to simulate before life existed, right? So that's all we got today for the origins of life. Next time on Wednesday, we're going to look at the evolution of life, the very early evolution of life, how we got from like single-celled stuff all the way up to like multicellular organisms. And the last thing is obviously the disclaimer here. So yada, yada, yada. That's all I got for today. So hopefully you guys enjoyed that. And just make sure to keep up on the discussion board. I posted some stuff up there for discussions. And if anything changes with your situation, let me know. Have a good one. Bye-bye.